Now that we know something about the structure of DNA and the structure of protein, we need to talk about their relationship with one another. We've already said that DNA is a recipe book for how to make things in the cell. And so as we look at proteins and how those proteins are put together, we know ultimately the recipe for making a particular protein is going to be found in the DNA. The central dogma, which was dubbed by Francis Crick back in the 1950s, helps us understand how we can start with our recipe that is in our code in DNA and actually go through the process of making a product, and that product is going to be our protein. So right now I'd like to just do a big picture overview of how we start at DNA and ultimately make a protein, where this happens in the cell, and some of the important steps that take place. And then we'll go through some of the details that we have of that. What you'll notice in the middle of this figure is something that we haven't spent a lot of time talking about, and that is RNA. We'll get to that here in a little bit. What we notice is that DNA, which is located, as we know, in the nucleus, is going to serve as a template for making this intermediary here, which is RNA. The step by which we go from DNA to RNA is called transcription. If you think about that word transcription, you can see the root transcribe. If you missed class and asked to get together with a classmate and you transcribed their notes, you'd essentially be getting a copy of their notes, but in your own handwriting. It would contain the important information. And that's what we do in the process of transcription. We're going to use DNA to get the same information now in a copy of RNA. This process of transcription takes place in the nucleus because that's where the DNA is located. Once we have a strand of RNA, then we go through the process of translation. And when you think about what that word translation means to you, translation means to take something from one language to another language. And that's essentially what we're doing when we start with RNA and use it to make a protein. If we think about this from a language perspective, both DNA and RNA are what we call nucleic acids. Their building blocks are nucleotides. And we've talked about the structure of DNA nucleotides, and we're going to review that here in just a second. But when we move from RNA to protein, we're going to a new language. Protein, of course, is like its own language, and its building blocks are in amino acids, the order and sequence of the amino acids. So in translation, we'll have to find how we can go from the language of nucleic acids to amino acids. And just like you might if you were trying to translate something from one language to another, we're going to have to consult a dictionary and look up what does a code in RNA translate into when we look at the specific order of the amino acids. Translation is going to happen in the cytoplasm. And specifically, it's going to happen at the ribosomes. We've talked about ribosomes before. Many of them are stuck to an organelle that we've discussed called the rough endoplasmic reticulum. And so that's where the process of translation will actually take place. So this RNA piece that we have here in the middle then is going to be formed in the nucleus, but then it's going to have to travel out through a nuclear pore and make its way to a ribosome, and that's where the process of translation will occur. Because RNA seems to play such a crucial role, we need to take a look at ways that an RNA nucleotide or the structure of RNA is different from DNA. When we look at our DNA and our RNA, I hope we remember that DNA is made out of nucleotides and that each nucleotide in DNA has three parts to it. It contains a phosphate group, it contains a sugar, and in the case of DNA, we know that sugar is deoxyribose. And we also know that there are nitrogenous bases. And there are 4 different ones in DNA: G, C, T, and A. So those are our 4 different nitrogenous bases. RNA nucleotides are very similar. They also have a phosphate group. They also have a sugar, But in the case of RNA, the sugar is ribose instead of deoxyribose. In fact, you can see this is how RNA gets its name, for its ribose, whereas DNA gets the D for deoxyribose. There are also nitrogenous bases. However, there's one change. There are G's in RNA, C's, A's, but instead of T's, there is U, and the U stands for uracil. So this is one of the differences that we have in the structure of RNA. So if we fill in our chart here, G, C, T, and A, those are the options for our different nucleotides. Here we would include G, C, A, and U. You may want to put a note to yourself, no T. Finally, we can always identify DNA from RNA in that DNA is a double strand. We've talked about it already, the fact that it is a double helix, and that those two strands are held together by hydrogen bonds. RNA, on the other hand, is just a single strand, and that single strand can be spread out like a long thread, or that single strand can be folded in upon itself and make a variety of shapes as well. In fact, when we look at RNA, there's many different kinds of RNA, and I'd like to mention a few of them here. Some of the ones that will become important are mRNA. M stands for messenger, so there's messenger RNA. There's a type of tRNA, transfer RNA, whose job it will be to transfer amino acids as we're going through the process of translation. There's rRNA, and the little r here stands for ribosomal RNA. Ribosomes are actually made out of RNA. And finally, while we won't discuss it much right now, there's RNA called microRNA. So while we look at RNA, we're going to have to distinguish between a number of these different types when we go through the process of transcription and translation. As we follow the flow of information from DNA to RNA to protein, we'll need to go through the details of these processes. We remember that our first step as we move from DNA to RNA is called transcription. In transcription, we're going to use DNA as a template to make a strand of messenger RNA. And remember that because DNA is in the nucleus, the process of transcription is going to take place in the nucleus. So let's look at this with a little bit more detail. Transcription falls into three main steps, remembering that this is a process. And the very first step is called initiation. So step one is initiation. Initiation is simply starting the process or initiating the process of transcription. You'll notice in this picture in blue is the DNA, and then during initiation we have to do something important to the DNA. We actually have to unwind the double helix. Now, we are not going to be transcribing the entirety of a cell's DNA in the process of transcription. In transcription, we're only going to be taking a section of DNA, just one gene, and going through the process of transcription. So you'll notice that our DNA is going to unwind, not everywhere, but just as we move along that section of DNA. We also have to have an enzyme that's going to make sure that this process occurs correctly. And the enzyme involved is going to be RNA polymerase. We've already talked about a polymerase before. Remember, DNA polymerase was the enzyme in DNA replication that made more DNA. RNA polymerase is the enzyme that we have in transcription that's going to make for us a strand of messenger RNA. During initiation, our RNA polymerase then has to attach physically to our strands of DNA, and not only is our double helix unwound, but then the hydrogen bonds that were holding together these two strands are going to have to be broken in this area right here. You'll notice that there's a much wider stretch between our two blue strands. Oftentimes textbooks will call this a transcription bubble. And this transcription bubble is where our RNA polymerase is located, and our DNA double helix has been not only unwound, but the hydrogen bonds have been pulled apart. What we're going to do is use DNA as a template for making a strand of RNA. However, we're not going to use both strands. What you'll notice here is the bottom strand is going to be used as our DNA template strand. Other textbooks may call this the sense strand, but that's a strand that the RNA polymerase is actually going to read to get the code and thereby use that to make messenger RNA. What that means is that our other strand, the top strand in this case, is our DNA non-template strand. The DNA non-template strand will not be transcribed. It's essentially not being used in this process at all. However, once transcription is over, these two sections of DNA, both the template and the non-template strand, will go back together, they'll reform their hydrogen bonds, and the DNA won't be damaged or changed during this process of transcription. Another important question we have to ask is, how does RNA polymerase know where the start of the gene is? And what you'll see here on the bottom strand in red is something called a promoter. The promoter is a DNA sequence that essentially is the beginning or the start of a gene. And this is where our RNA polymerase is going to attach. So in the stage of initiation, our DNA is going to unwind, our hydrogen bonds are going to break, and our RNA polymerase is going to attach to DNA at the site of the promoter. The second step is called elongation. Elongation is where RNA polymerase gets to work adding in complementary base pairs of RNA nucleotides as it reads the DNA template strand. So let's make sure we have that down. RNA polymerase is going to be adding RNA nucleotides. And it's going to do that as RNA polymerase is reading our DNA template strand. Let's actually look more closely at this process. I want to show you a little bit more detail. This is a picture out of your textbook, and you'll be able to see it there. And we can see something very similar to what we've started with. In this scenario, again, our bottom is going to be our template strand. We can see our RNA polymerase here, and we can see our blue DNA has been separated. The direction of our synthesis is this direction. RNA polymerase is moving, wobbling along our DNA, and as it's doing that, this is unwinding as it moves that direction, and then this is going to reform our double helix with our hydrogen bonds after RNA polymerase has passed over it. As RNA polymerase reads that template strand, it's going to put in the complementary RNA nucleotide. So let's pick up right here and see what RNA polymerase is doing. If there's an A in DNA, the complement to that is going to be U in RNA. Remember that U is taking the place of T. Remember that U, uracil, is taking the place of T. There's no T in RNA. If there's an A in DNA, there's a U in RNA. If there's a G in DNA, there's a C in RNA. So you'll notice that these are the complementary bases that are forming between our DNA and our RNA. And temporarily, they'll actually be connected to one another by hydrogen bonds. You can see that our growing strand of RNA is shown in that pink color. And so here is our growing strand of RNA as our RNA polymerase will move along. That's essentially what's occurring during elongation. The third step is termination. Termination occurs when RNA, as it's been moving along the template strand, reaches a series of bases on DNA that indicate the end of the gene. And much like our promoter did at the beginning of the gene, this is going to be the signal for RNA here at the end to detach. So at this point, RNA polymerase detaches from the DNA. And at that point, we have three things that we need to consider where they're headed. First of all, our DNA is simply going to rewind and go back to the way that it was. Remember that DNA hasn't been used up or damaged in this process. We just used it as a template. Much like when you use a recipe at home, it's not as though you tear that recipe page out of your cookbook and throw it away. You just put it back so that you can get it out and use it again. And this gene might be transcribed again, maybe in 5 minutes and maybe next month. Our RNA polymerase, in being an enzyme, can also be used again. We remember that enzymes don't get used up in the process of their reactions, and so RNA polymerase could be used again. And then finally, our RNA, our strand of messenger RNA, is the thing that we're most interested in. And we know that that's going to actually leave the nucleus and head out into the cytoplasm, and there it's going to be involved in the process of translation. Once our strand of messenger RNA has been made, we will refer to that as the primary RNA transcript. So you may hear that in other videos or in your textbook. And if you look closely at this primary RNA transcript, you'll notice that it has these terms exons and introns, and you can see that they're alternating as it makes its way along. What we find is if we had started with DNA, which we did, and we went through the process of transcription to make this transcript, transcription took everything that was in our DNA gene and transcribed it. However, not everything in that gene is going to be absolutely necessary for us to make our proteins. So what we want to do is clean up the messenger RNA a little bit. We want to go through what we call messenger RNA processing and really neaten up that particular strand of messenger RNA. When we look at exons, exons are the pieces that we're actually most interested in. An exon is essentially our protein coding region. And those protein coding regions are what we want to gather from our RNA transcript and send to our ribosome so we can tell it specifically what to make. We can see that our exons then are present in our RNA transcript, and then after we go through this processing, we've put all of the exons together. You'll also notice that we've removed some things, and what we've removed are these introns. Introns are called intervening sequences, and they are not going to contain information that's directly related to how to make our protein ultimately. So we know that while they're present here in our primary RNA transcript, these are going to be removed, and you'll notice that we don't have any introns here once we've gone through messenger RNA processing. So that's one part of messenger RNA processing, is to remove the introns and to splice together. So that word splice means that we're going to essentially glue these exons together here so that we've got nice, clean messenger RNA. One other thing that we're going to add to our messenger RNA is a cap and a tail. You'll notice that at this end we've added what's called a 5' cap. And here at this end, we've added what's called a poly-A tail. We're going to add these things in order to prevent degrading of our messenger RNA. Remember, our messenger RNA is going to be leaving the nucleus and making its way through a nuclear pore and going all the way to a ribosome. We don't want any of the information in our exons or any part of our spliced RNA to get damaged. We don't want to shear off any important nucleotides that are there. So this is going to act like some protection to make sure that we contain and keep all of our messenger RNA together. In general, that's the purpose of our messenger RNA processing. We want to make sure that we have some stability for our messenger RNA and that we're preventing degrading of it. We also know that it can aid the transport of our messenger RNA out of our nucleus and toward our cytoplasm. Once our messenger RNA leaves the nucleus, goes to the cytoplasm and to a ribosome, we're ready to undergo the process of translation. There are some other parts, some pieces, some machinery that we have to include in the process of translation. So I'd like to cover those here before we put all these pieces together. First of all, we notice that we've got our messenger RNA strand here, and we already know the backstory of this. This was made by the process of transcription back in the nucleus using DNA as a template, and we know that it got processed and that it came to our ribosome. This messenger RNA then is carrying the information that was on our original DNA gene, and it's bringing it here to the cytoplasm with the instructions, really, of what type of protein to make. What we see in blue in this picture is the ribosome. So the ribosome actually has two parts to it. Books will call the larger part a large subunit, and that's just the bigger part of our ribosome, and there's also a small subunit. And these two fit together a little bit like a shoebox. And it's right where they meet, where our messenger RNA is going to thread through it, and where our third component of our machinery is going to come into play. And that's going to be a transfer RNA. We've mentioned transfer RNA before, but here we're getting a good look at it. A transfer RNA does just what its name suggests. It's going to transfer or deliver the amino acids to this area, and they're only going to be able to drop off their amino acid in a particular order, thereby growing the protein in correspondence to what is on the messenger RNA strand. Transfer RNA is also made out of RNA. I wanted to just draw you a simplified picture of what a transfer RNA would actually look like. So if you could imagine a transfer RNA simplified, on the top part, this is where we have the amino acid. So we remember, I hope, that there are 20 amino acids, 20 different amino acids. And that means that a transfer RNA is only going to carry one kind of amino acid. They're very specific. On the other end of a transfer RNA is going to be a code that ultimately is going to have to match up with our messenger RNA. And this code that's going to be on the bottom portion of our transfer RNA is called an anticodon. Anticodons are going to help the transfer RNA make the correct match with the section of messenger RNA. Because we are going through the process of translation, as I mentioned earlier, we have to be able to look up in a dictionary how we translate something from RNA to actually translating it into protein. And so for that, we're going to look at what we call our genetic code. This is actually the dictionary that links up RNA nucleotides with our amino acids. What we're actually looking up here are what are called codons. So I need to make sure that we define the word codon, and then we'll go back and look and see how anticodons are going to play a role. If we had a section of messenger RNA that, for example, was AUGGUC, I'm just going to make a section here of our messenger RNA. So that's our messenger RNA. What we find is that in order to figure out what amino acid is going to be in order according to this code, we couldn't use just one letter to equal one amino acid. Because if we did that, we only have A, G, U, and C, only 4, and that means we'd only be able to code for 4 amino acids. And we couldn't use a 2-letter code. Instead, we're going to have to use a 3-letter code. We're going to read RNA in sections of 3. So we're going to break the messenger RNA into groups of 3, and each of these groups of 3 is called a codon. So codons are found on messenger RNA, and we're going to look these codons up in this right here, which is our codon dictionary, to find out which amino acid is going to go in which order. Let's take a look at our codon dictionary to learn some things about it. What you'll notice when you look at the codon dictionary, I hope, is that you can always find easily your first letter, your second letter, and then your third letter and find the particular code. You'll also notice that each of these codes is going to code for a particular amino acid. So you'll see, for example, right here we have a code CCA. That's a codon on messenger RNA, and it's going to go with the amino acid pro, which is for proline. So all of these things here are going to be amino acids. Whereas any code that we have here with GUG, GUA, all of these things here, those are going to be the messenger RNA codons that we get from our messenger RNA strand. Let's notice some other things about our dictionary. Our dictionary displays what we call redundancy. And what that means is that for each amino acid, there may be more than one code that codes for it. So let's pick an amino acid. For example, let's look at proline again. There are a number of ways that you could get proline as an amino acid in your polypeptide chain. You could have a CCU, CCC, CCA, or CCG. All of those codons would lead to having the amino acid proline. There are a couple exceptions. One is that there has to be what we call a start codon. AUG is always the start codon. Translation won't really begin until we have an AUG in the messenger RNA, and that is always going to code for MET, and MET stands for methionine. That's the very first amino acid that we'll have in all of our proteins. How do we know when the process of translation will be over? Well, you'll notice that we also have some stop codons. UAA, UAG, and UGA are all stop codons, so that when we reach that section on a strand of messenger RNA, the process of translation will stop. So let's do this example that we have together and try to find the corresponding amino acid. As you might expect, our first section here is AUG. That's our first codon, and that's what we'll look up here. And that first codon is going to correlate to the amino acid MET. Our second codon on messenger RNA is GUC. So we come here to G, then we find our U, then we find our C. And there is that codon, and it's going to correspond to the amino acid valine. And so we'll put val there. Our next codon is GAU. So when we look that up in the codon dictionary, we start here with G, go over to this box here, that's where our A's are, and we can find GAU right here, corresponding to the amino acid ASP. And then finally, we have UGA, and UGA we can see here is a stop codon. And so the process is going to stop. We, in this particular example, have 3 amino acids, MET, VAL, ASP. We're going to link these 3 together with a peptide bond, thereby making our protein. This is how we're going to use this codon dictionary to look up messenger RNA codons to determine which amino acid is going to be present. Let's put all of our machinery and pieces together to look at the entirety of the process of translation. In this picture here, which is out of your textbook, let's start on the top panel and find out how this process occurs. First, we can identify our strand of messenger RNA. That's here in that orange color. We can also see our codons, and so it's a good idea to block these off in groups of 3 so we can identify those codons. At this point in the process of translation, we will be using the same 3 terms that we used in transcription. The start of translation is what we call initiation. During initiation, the small subunit of our rRNA, so part of our ribosome, is going to receive our messenger RNA, and you'll be able to see that our first transfer RNA is going to arrive bringing in our first amino acid MET. This is where I want to be really clear about our codons versus our anticodons. Remember that on our messenger RNA, these things here are codons. And the question is, well, how does the correct transfer RNA know that it's the next one in? Or how is it allowed to dock here at the ribosome and actually drop off its amino acid? Remember that on transfer RNA we have an anticodon. And the anticodon is going to be the complement in RNA to our codon in messenger RNA. Let's make sure this is correct. A in our codon is going to go with U in our anticodon. U goes with A, G goes with C. So the transfer RNA that's carrying MET has to have the anticodon UAC. In that way, the UAC is the anticodon to the codon and messenger RNA, and our transfer RNA is allowed to dock and will be allowed to drop off the MET, our first amino acid in this polypeptide chain. Remember that on the previous dictionary, what we're looking up are not anticodons. We always look up codons. So don't get confused about that. It's important to remember our dictionary is a codon dictionary, not an anticodon dictionary. Let's get a preview then of what you think our next amino acid in place will be and which transfer RNA will bring it in. Our messenger RNA is UUU. This is our next codon after AUG, and that's what we want to look up in our dictionary. So why don't you pause the video for a moment, look up UUU, and find out which amino acid will go with it, and then you can hit play again. Did you find it? I hope you've found PHE. That's going to be our next amino acid. And if we've got a transfer RNA that's going to be carrying PHE, let's sketch that here off to the side. That's the one we expect next. And then let's also figure out the anticodon that we expect to see on the transfer RNA that carries this particular PHE. If we have UUU, then the anticodon is going to be AAA. So let's look at our next panel and see if we're correct. You can see that we were correct. The next transfer RNA that came in was bringing the phenylalanine, PHE, and it had the anticodon of AAA that went with the codon UUU, which was on messenger RNA. I don't want you to worry so much about the P and the A. They essentially are like a dock or a gate in an airport of the position of these transfer RNAs, but that's a little more detail than I wanted to cover. All right, let's do it again and see if we can predict our next transfer RNA and our next amino acid. You can see our next codon is going to be CGA, and that's what we want to look up in our codon dictionary. Why don't you pause the video for just a moment and see if you can find that amino acid. Did you find it? I hope you found that it was ARG. So if we were to draw the next transfer RNA that's going to be coming in after the one carrying PHE, our next one will carry our ARG, and let's find out what its anticodon should be. If we have CAG in our messenger RNA codon, then C goes with G, G goes with C, and A goes with U. So this should be what our anticodon should look like for the next one that's coming in. Let's see if we got it right. We did. We can see that this transfer RNA right here is bringing in the amino acid ARG, and that the anticodon is GCU. The process by which new amino acids are delivered by the transfer RNAs and placed in position according to the anticodon and codons matching up, that's really our second step. And our second step was elongation, where our amino acids are being added and our polypeptide chain is getting longer and longer. Our third step, similar to the terminology we used in transcription, is termination. Termination is going to occur when we get a stop codon that's going to be present in our messenger RNA. And when that occurs, we'll find that the whole process is going to stop. It's going to terminate. And that's when our large and small ribosomal subunits will break apart. We'll be able to see our messenger RNA now free, no longer being translated, and that we'll have our finished polypeptide chain. Once we have our polypeptide chain, we consider the process of translation to be over. That was the goal of this entire process, was to use DNA to make a polypeptide chain, and that's our protein. Why don't we go through another example together so that you can get the hang of how translation occurs. This is an example out of your course manual. Let's first block off our codons in our messenger RNA. And if we go back to right here, this is where we will start with a start codon. And then we have CUA, UAU, GGU. We're really picking up in the middle of translation, in the middle of elongation right now. I hope we can see that we have our growing polypeptide chain, and these should match up very nicely to our codons that are here on our strand of messenger RNA. So let's make sure our figure is correct. If we had AUG, that was going to be the codon that brought our MET. We could look AUG up in the dictionary and we would find MET. Now let's find our next codon. Our next codon is CUA. Why don't you pause the video for just a minute to see which amino acid CUA will code for. CUA is going to code for leucine, LEU. Our next codon on messenger RNA is UAU, and when we look UAU up in the codon dictionary, we'll see that it goes with TYR, that amino acid. In fact, we can see this transfer RNA having just dropped off the TYR. Let's make sure that they got the anticodon correct. So if we have in our messenger RNA our codon of UAU, what should be the anticodon that we have with transfer RNA? U goes with A goes with U goes with A. So that's correct. Let's look at the one that's in position now. Our codon is GGU, so let's look that up in the dictionary. That should go with GLY, G-L-Y. And our anticodon on our transfer RNA should be the complement. G goes with C, G goes with C, U goes with A. There is a mistake in our next panel. Let's see if you can find it. Let's look at our next codon that's coming up, AUC. And if we look that up in our dictionary, we're going to find that it does not go with iso. The amino acid should be ILE. So please make sure you make that correction. Let's take a look at our anticodon on transfer RNA and make sure that that's correct. A goes with U goes with A, and C goes with G. Remember that once this process hits a stop codon, this entire process will be terminated. Now that we've covered the details of transcription and translation, I hope that you could go back and look at these three things in order again. Here we see the process of transcription. That occurs in the nucleus, where we use DNA as a template to make a strand of messenger RNA. Then we went through the process of processing RNA, where we removed our introns, we spliced our exons together, and we added a cap and a tail. And so that process of RNA processing happens in the nucleus as well. And then finally we can see the process of translation that occurs in the cytoplasm at a ribosome, where our transfer RNAs are bringing in our amino acids and allowed to drop them off, assuming that their anticodon matches the codon on the messenger RNA.